Yes, Dr. Junaid.
So should we start?
Yes, sir.
Good morning to all. I warmly welcome all of you to today's informative webinar. My name is Junaid Tantre. As a co-convener of this event, it brings me great joy to see wide-ranging and active audience participating online. Your attendance not only enhances our dialogue, but also highlights our shared dedication to progressing in our respective fields. I'm truly looking forward to engaging in thoughtful cooperation discussions as we commence this educational journey collectively. SPSR is renowned for its engaging webinars, strengthening its reputation as a top choice for learning and growth in the pharmaceutical sector. I trust today's webinar will uphold its standard, presenting valuable perspectives from respected speakers. Let's embrace this chance to gain knowledge, participate actively, and further our profession. Welcome, and I expect a fulfilling experience for everyone involved. Thank you for your participation. Today marks an important occasion as we convene for the 55th SPSR webinar titled Multiomics Approach, a Paradigm Shift in Drug Discovery. This webinar is a part of prestigious series organized by Society of Pharmaceutical Sciences and Research with Dr. Shashi Alok sir, the National President of SPSR, providing respected mentorship. With his leadership, SPSR remains instrumental in promoting progress and creativity within pharmaceutical sciences. The SPSR is committed to the progression and enhancement of pharmaceutical field through diverse strategies. We aim to facilitate conferences that promote pharmacy-related initiatives and endorse programs and activities addressing public health issues in India, with an emphasis on disease prevention. We also make it a point to acknowledge and celebrate pharmacists whose significant contributions inspire further advancement in our field. Central to our purpose is the commitment to fostering the growth and spread of pharmaceutical knowledge, ensuring the consistent and rich engaging of information regarding pharmaceutical practices and services. By pursuing these goals, we strive to raise both the quality and influence of pharmacy profession, cultivating a proactive and cooperative community. It's a great privilege to introduce Dr. Dharmendra Kumar Khatri, sir, Associate Professor at NIMS Institute of Pharmacy, NIMS University, Rajasthan, Jaipur, as a distinguished speaker. Dr. Dharmendra Kumar Khatri, sir's expertise is a significant asset to our event, offering depth and understanding in the field. His contribution promises to enhance our conversations and deepen our examination of multi-omics approach advancements. We are thankful for his involvement and expect an informative session. We are honored to welcome Mrs. Monika Sabharwal, ma'am, as the convener of this webinar, National Secretary SPSR and Managing Editor of IGPSR and IJP. Her extensive contributions and leadership reflect her dedication to pharmaceutical sciences and research. We are very fortunate to have her expertise steering our session today. I am pleased to invite Mrs. Monika Sabharwal, ma'am, to warmly welcome our distinguished guest speaker, Dr. Dharmendra Kumar Khatri, sir. Ma'am, over to you.
Thank you so much, Mr. Julian Tantre. Hello, everyone. I, Monika Sabharwal, have a distinguished privilege to welcome you all to 55th SPSR webinar on multi-omics approach, a paradigm shift in the drug discovery, hosted by Society of Pharmaceutical Sciences and Research (SPSR). This event represents another landmark achievement as we continue to progress in the field of pharmacy. We are privileged to welcome Dr. Dharmendra Kumar Khatri, sir, Associate Professor at NIMS University, Jaipur, Rajasthan, India, as our distinguished guest speaker for today. An expert in pharmacology research and academia, Dr. Khatri's expertise on the multi-omics approach to drug discovery is eagerly awaited. The topic of today's webinar, Multi-omics Approach, a paradigm shift in drug discovery, is particularly relevant as it explores how integrating various omics technologies can revolutionize the way we discover and develop new drugs. This approach promises to enhance our understanding of disease mechanism, identify novel therapeutic targets, and ultimately improve the patient outcomes. We are pleased to share that we have received over 2,000 participants from 20 countries, highlighting the worldwide relevance and the significance of today's topic of discussion. Today, we are also celebrating a milestone, the 55th webinar on SPSR series. Our journey has been marked by incredible support from participants around the world, and we are deeply grateful for your positive feedback we continuously receive through the various platforms. This feedback not only boosts our morale, but also drives us to keep improving and offering valuable content to the pharmaceutical community. In this year alone, the SPSR webinar series has reached an impressive milestone of more than 50,000 registrations. This remarkable achievement is a testament to the hard work and dedication of our SPSR team. We strive to provide the best content and learning experiences through our webinars. And regular attendance of our audience on every Sunday reflects the positive impact we are making. We are pleased to warmly welcome Dr. Shashiyala, the esteemed National President of SPSR. His wise leadership has caused a remarkable transformation within SPSR, making it a model of excellence in the pharmaceutical sciences. I would like to express my sincere gratitude for invaluable contributions of Mr. Junaid Tantre, the co-convener of this webinar, as well as the organizing committee members, Dr. Pradeep Gulani, Dr. Sanjay Nagdev, Mr. Ankur Agarwal, and all other SPSR team members. Their tireless commitment has been crucial in making a vision this event to a reality. The seamless execution of this webinar is a testament of extraordinary effort by our team. Their steadfast drive for perfection and dedication have been pivotal in ensuring the event's triumph. I wish to express my profound appreciation to each member of SPSR team for their rigorous focus on specifics and their consistent enthusiasm that brought this webinar a future. My sincere thanks to all of you for your active engagement and significant contributions to today's webinar. Your involvement drives our shared commitment to advancement in the pharmaceutical sciences. I trust that our conversations today will provoke you to be informative and motivational, making a lasting impact on our career paths. I have a very great news for all of you. We are thrilled to announce that we are launching an e-poster competition for our pharmaceutical students. This is a fantastic opportunity for all to showcase your innovative research and ideas in the field of pharmaceutical sciences. The competition will be held during the upcoming live webinar. We encourage all students to participate and present their work in the digital format. This is not just a competition, but a platform to learn, share, and grow together. So, let's embrace this opportunity to shine a light on your hard work and creativity. Students will get updates on our various social media handles, and I request you to stay updated on them. We look forward to see you for an insightful contribution. Before I wind up, let's keep at the forefront of innovation with SPSR. Subscribe to our YouTube channel for exclusive webinars, professional insights, and the latest research in the pharmaceutical sciences. Become a part of active community that will shape the future of healthcare and the pharmacy sector. Keep up to date by subscribing to our YouTube channel and turning the notification bell icon for immediate alerts. Follow our progress in advancing the pharmaceutical sciences that will benefit the society overall. Without further ado, let's welcome Dr. Dharmendra Kumar Khatri, sir. Thank you for joining us today, and I look forward for the enlightening session. Over to you, Mr. Junaid Tantre.
Thank you for the words of appreciation, ma'am. I appreciate Mrs. Monika Sabharwal, ma'am, granting me the opportunity to present this webinar. And now we will start a short introduction to the guest profile. Dr. Dharmendra Kumar Khatri, sir, is an accomplished associate professor in the Department of Pharmacology at NIMS University, Jaipur. Sir previously contributed to Department of Biological Sciences at NIPER, Hyderabad, India for almost 5 years. Dr. Dharmendra sir holds a Master of Pharmacy and a PhD from esteemed Institute of Chemical Technology, Mumbai, formerly UDCT, and has been a recipient of University Grant Fellowship for Junior Research Fellow twice and Senior Research Fellow during his postgraduate and doctoral studies. As a young neuroscientist, Dr. Khatri sir's focus on leveraging multiomics, bioinformatics, genomics, proteomics, metabolomics, and lipidomics to identify precision targets and refine fit molecules with a particular emphasis on understanding alpha synuclein dysregulation in Parkinson's disease at post-transcriptional and post-translational levels. Dr. Dharmendra sir has an impressive academic record, having published over 120 articles in peer-reviewed journals, authored 2 books, contributed to several book chapters, and holds 4 patents. His mentorship has guided more than 30 masters and 6 PhD students. His outstanding contribution to neuroscience have earned him numerous prestigious awards and fellowships, including Associate Fellowship of Telangana Academy of Sciences, the Excellent Oral Presentation Award at 43rd Annual Scientific Meeting of Korean Endocrine Society, and the Young Neuroscientist Award from Japanese Neuroscience Society. He is also honored with the first position for Dr. P.D. Patil Best Thesis Award 2023 in the subject category M.Pharm Pharmacology as a mentor. In addition to his research and teachings, Dr. Dharmendra sir is an ambassador for the International Society of Neurochemistry for the year 2022 to 24 and 2019 to 21, and has been recognized by Osaka University with the Next Generation Young Neuroscientist Award in 2018. Dr. Dharmendra sir has received the Lalita and Ravindranath International Travel Award to present his research at the American Society for Neurochemistry meeting in Montreal, Canada, 2019, and the Best Poster Award at the 4th International Conference of Korean Movement Disorder Society in 2023. Dr. Dharmendra sir serves on the editorial boards of several distinguished journals, include Molecular Neurobiology, Journal of Alzheimer's Disease, Annals of Neuroscience, Frontiers in Neuroscience, and Vino academic journals. He is an active member of various esteemed scientific societies such as the Asia Pacific Society for Neurochemistry, Japanese Neuroscience Society, International Society of Neurochemistry, International Parkinson and Movement Disorder Society, Indian Pharmacology Society, Indian Academy of Neuroscience, Society for Neurochemistry, India, Association
Hey,
of
Janet, fine.
Physiologists, and
Let us
Pharmacology
start.
of India. So we are pleased to announce that over 2,000 attendees have registered from around the world, representing a variety of nations. Notably, we have substantial participants from countries like Nigeria, India, Algeria, Bangladesh, Indonesia, Kenya, Malaysia, Syria, UAE, UK, Vietnam, and Yemen. So thank you for all joining us today. Let's make this webinar insightful and enriching. Over to you, Dr. Dharmendra sir.
Good morning, good afternoon, good evening. Considering the multiple participation from the across the world, so I don't want to stay as a good morning because here the time is 11. So probably people who are sitting on the overseas may be having a different time. So for everyone, afternoon and evening. First of all, I would like to thank the SPSR organizing committee, especially Dr. Alok, Dr. Junaid, and Dr. Monika for giving me this opportunity to showcase some of my research work as a part of, to educate my own society, which I belongs to. And it gives me immense pleasure to, give me something back to my own young generations of, the pharmacist. It is also a privileged movement for me. I have been informed by the organizers that this is the first time that we had a highest participant from the highest number of countries, and that too in a very short notice. And further, without the delay, I would like to start my talk. So, can you somebody confirm? Is this visible?
Yes, sir, it's visible. Can you please put it in presentation mode?
Is it?
Yes, sir. It's totally. Yes, sir. Please proceed, sir.
So since the multiomics is not a very small terminology, it's a very big terminology, and it is a vast, it's an ocean of, topic which won't be ended in just 1 hour. It takes a lot of expertise. So I'm having expertise on certain parameters, which I have been doing with my own research group, and we have been doing those as a part of our target, which we wanted to refine in case of Parkinson's disease. So I'll give you very first, in initial slides, a bit background about what exactly is the multiomics. And then I'll move forward to my specific target, what we have been doing as a part of our research. Hope my slides is moved. Can you confirm?
Yes, sir. It's moving.
Okay, fine. So if you look at the typical and the conventional drug discovery process, which start from the target selections, and then we as a scientists are going to validate this target, and eventually we take it further for this target to find out the lead molecules, where we do the lead discovery utilizing multiple platforms. And eventually we wanted to find out a drug which is most suited with the aim of having a least toxic, higher efficacy, and most safest compound. So these are the three endpoint that is associated with any molecules that we wanted to take further for our clinical trial. Should have it. First is the safe and does not have any toxicity, and of course the efficacy is very important. However, during the lead discovery process utilizing multiple platforms, if the molecule is found to be very promising, but however it is having a good amount of, or maybe a huge amount of side effects, then the medicinal chemist comes into the picture, and they help us in modifying those molecules by doing the SAR studies on the side chains, and they try to develop a molecule which fits most and which suits most for the further steps in preclinical drug discovery. And further, when we perform the very specialized and specific in vitro test using the targets, especially enzymes, maybe the receptors, or maybe the ion channels or transporter. So we use a very simulated conditions in case of using cell culture, so maybe the ex vivo approach, and then we try to see what the candidate behavior, especially in vitro conditions. So finally, once we get the data from in vitro, which is eventually going to get translated in vivo settings, where we use a most suitable disease model, and then we try to find out varieties of approaches, again utilizing moleculars, especially genomics and proteomics, and finally those lead compound which is being refined from almost all these steps eventually being taken for the clinical trials. So I'm not going to focus on the clinical trials, while I'm going to focus on first and second steps, and finally on the fourth or fifth steps. So if you look at a typical approach that we employ, especially in the target selection and the validation of the target, which what we have been taught or what we have been thinking of conventional drug discovery is now no longer exist, because of evolvement of the scientific platforms and technologies has made us to think about the multiomics approach, where we can have a very defined target, which is precision based target. So in those cases we use multiple approaches, and I have been using, and I'm going to show you a few data that how we are using the bioinformatics approach, where we are trying to extract few of the genomics data which are already published, and we try to make the network pharmacology, gene ontology networks, or gene co-expression networks, and try to find out which target could be a most promising for one particular disease. But in this case, I'm going to focus only on Parkinson's disease. And we have multiple other data, which includes whole genomics or maybe the genetics data. And then the lead discovery based on those targets, you eventually going to go for the high throughput screening based on the, again, software based, or you can do the in vitro test as well. So when I say high throughput screening on the software basis, where you have a multiple libraries available, I will say that a typical example for that is the Campbell. So you can use millions of compounds using your target structure, especially of receptor, whatever it is, and you can construct your target and your lead compound, your screening compounds of millions of library. You can actually, have them interacted, and you can refine based on the certain, parameters, which eventually I'm going to discuss in the further slides. And then you, if required at all, you can develop your own assays, and then further those refined molecules, which comes from the lead discovery, eventually get modified with the help of medicinal chemist people, where they do some modifications using the SAR in order to make a as I mentioned, the most suitable compounds. And then we make a varieties of cellular models to find out the selectivity in order to finally make a refined lead molecules, which gives us a promising results to eventually get translated into the in vivo settings. And those lead molecules, which are refined lead molecules, eventually being used with the most suitable animal model of disease models. And there are three important approaches which we or everybody does is actually behavioral studies, biochemicals, and molecular studies in order to find out whether the drug is acting on the right target or not, or if it is acting on multiple pathways, how it is going to act. And finally, it is being done in the human clinical trials. So my talk will be focused initially on the target selections and the lead discovery. So I'll be having two important aspects in my presentation. So one is called as target-based drug discovery, and one is called a lead-based drug discovery, hit-based drug discovery. So on one side, I will have a target fix, and I wanted to find out the new molecule. And at one side, I have a molecule with me, and I wanted to find out the target. So this is called as hit-based drug discovery, and one is called as target-based drug discovery. And I'm going to discuss with you guys in the next slides. So just for the background information, that there are plenty of new technologies are emerging in order to find out a very precise drug targets, which are not limited to on every step of the drug discovery. There are multiple platforms out there. We are using bioinformatics approach. We are also using the different tools in order to design the drug compounds. We are using the library also. We are doing the docking. We are also doing the QSARs. We are also doing in silico ADMET profile also in order to find out the drugs before actually it is being taken into the in vivo settings. We do certain software-based the ADMET profile as well. So even we can do certain toxicity profile also of those compounds using certain softwares. Overall, if you look at the what exactly is the multi-omics approach, which includes the varieties of layers of the data, which includes genomics, proteomics, compartmental, cell types, phenomics, pathology, metabolomics, and the different types of the drugs, which eventually make them into the network pharmacology, and you try to create a one specific pathway that this lead compound or my target is actually because when we talk about any disease, especially of most of the diseases are of chronic in nature, so they don't follow a single pathway. They are of multiple in origin. So if you look at typically in case of Parkinson disease, the example which I'm giving right now here, it is not only because of alpha synuclein mutations, it is also because of the LARC, GB, and Parkin mutations as well. But if you look at the downstream pathway that they follow, is not only limited to mitochondrial dysfunction, they do also have a certain toxicity as the chromatin level. They also have a lysosomal dysfunctions. They are also going to increase oxidative stress. They are also going to cause dopaminergic neuronal death. They are also going to disbalance the sphingolipids, which are very important. They are also going to disbalance the growth factors as well. So if you look at this, the algorithm of finding the exact mechanism of those data, it is very difficult. However, using those different platforms or computer-based platforms, we can actually refine our targets, and we can actually find out a very specific disease-specific targets, and then we can eventually make a very specific drug candidates as well. So I'll show you a few of the real-time data what we have done so far. Before going to talk about the what data we have been producing, I just would like to give you a one slide background about the disease, what I have been doing it. You guys may be knowing in case of Parkinson disease, comparing to the healthy brain, the PD patients have lost their specific brain area called as basal ganglia, and especially on the midbrain, this is called as substantia nigra, which is highly dense with the dopaminergic neurons. These neurons release a specific neurotransmitter called as dopamine, which eventually going to bind to the postsynaptic neurons. It performs functions. So the dopamine and acetylcholine both together maintains the body posture, especially, and the motor movement. However, in the disease conditions, like typically, which is what so far known in the scientific domain, that alpha-synuclein aggregations, which eventually going to get ubiquitinated and form the Lewy body inclusion into the intracytoplasmic inclusion, which makes these neurons vulnerable and makes them to die. That means that in the Parkinson conditions, those neurons are not sufficient to release the dopamine, and then it is going to cause the different kinds of disease symptoms. It is believed that in the very early stage, it is affecting the nigrostriatal pathway, which starts from the SNPC and it goes to the stratum. This is the pathway which is going to affect it in the very initial stage or mid stage of the Parkinson disease. And that's where we could see that the voluntary movement is going to get compromised. And that's where the first line of treatment for Parkinson disease comes as the levodopa as a gold standard in order to improve the motor movement. In the later stage of Parkinson's disease, the pathways that follows is mesocorticals, mesolimbic, and tuberoinfundibular, which is going to affect almost all part of the brain, which includes your motivations, cognitions, anxiety, and everything is going to come at the later stage of Parkinson's disease. So we are interested in finding out this culprit, how it is happening, and why it is happening. And I'll going to give you a few real-time data what we have been doing. So before going to get into the detail, I wish that my audience is supposed to see this video very carefully, because this video is very close to my heart, and what my research is focused on, everything this video has it. So kindly watch it. This is from the Roche, developed by the Nature.
Parkinson's is the second most common neurodegenerative disease after Alzheimer's. The most visible features of this disease are a slowness of movement, muscle rigidity, and tremors at rest. There are also many, often debilitating, non-motor symptoms, some of which may present before the motor problems. Parkinson's affects many areas of the nervous system and different types of neurons. However, much focus has been placed on neurons in brain regions associated with the overt motor symptoms, notably a region of the midbrain called the substantia nigra pars compacta. This region forms part of a major pathway in the brain that's critical for facilitating movement. In Parkinson's, dopaminergic neurons in the substantia nigra gradually die, leading to the malfunction of this pathway and the characteristic motor problems. Drugs that replace or mimic dopamine are often used to treat these motor deficits, but they grow less effective over time. Deep brain stimulation may also be used to treat symptoms. However, no current treatment slows the neurodegeneration. A distinctive pathology in most cases of Parkinson's is clumps of misfolded proteins within neurons. Lewy bodies are the most common type. A characteristic component of these is a misfolded protein called alpha-synuclein. These molecules can form small repeated units called oligomers, or longer fibrils. Mounting evidence indicates that these are toxic to neurons and play a key role in driving Parkinson's. Unwanted proteins are normally cleared by the cell's different types of protein-degrading machinery. Some evidence points to these systems being overwhelmed by misfolded alpha-synuclein, and this might be a key factor in what kills the neurons. Parkinson's has also been linked to problems with mitochondria. Mitochondria provide cells with the energy to perform vital functions. They're highly dynamic and can fuse together or break up into smaller versions in response to a cell's energy demands. They can also be transported to areas of a cell that need them the most. However, in Parkinson's, these processes can be impaired, and mitochondria are unable to sustain proper neuronal function. As they become old or damaged, mitochondria are removed and replaced. Again, this recycling is thought to be disrupted in Parkinson's, leading to the accumulation of damaged or worn-out mitochondria. Another idea is that the cells called glia surrounding neurons may play a role in Parkinson's. As dopamine neurons are lost, one particular type of glial cell, called microglia, is thought to take up the resulting cellular debris, triggering an immune response. Once activated, they release inflammatory cytokines, which activate neighboring microglia and another type of glial cell called astrocytes. Chemicals released by activated microglia and astrocytes have been shown to injure neurons. It remains unclear which mechanisms drive the disease process in Parkinson's. What is clear is that with time, more areas of the nervous system develop pathology. And one emerging idea is that this happens through the transmission of misfolded alpha-synuclein. While the multifactorial nature of Parkinson's disease progression might make it hard to fully understand, it does provide a number of potential therapeutic targets for slowing or even halting the neurodegeneration. For example, enhancing the clearance of abnormal proteins and blocking their transmission, improving the function of mitochondria, and targeting the neuroinflammation are all currently active areas of research.
Okay. So I have stopped with video and I came with the next slide. Is it visible?
Yes, sir.
Okay. So this is my target, and as you guys have seen in case of video also, this is the culprit which is going to play a great role in disease pathology. If you look at this alpha-synuclein, which is being transcribed by the fourth chromosome from the Q arm, which is here different ligases, during the transcription and translation, this particular gene called as SNCA is going to transcribe the SNCA mRNA, and this mRNA eventually going to translate the protein called as alpha-synuclein. This alpha-synuclein protein has a three component called as non-amyloid components, then carboxylic end, and the N-terminal end. And usually in all mechanisms, every protein has to go under the post-translation modifications, and these protein, especially alpha-synuclein, also goes under the varieties of post-translation modifications, and they do perform their varieties of functions on the varieties of macromolecules of the every cells. In this case, this alpha-synuclein has a great role to play, not only limited to mitochondria, it also has a great role to play on lysophagy, endoplasmic reticulum, and nucleus as well. This is the normal healthy state conditions. And during the Parkinson disease, what happened? This particular gene is going to have two checkpoints where it is going to get modified, either at the transcription level, where the miRNA dysregulation takes place, or either at the post-translation modifications, where a specific mechanism that involves in PTM going to convert this particular monomer, which is of native form, into the oligomeric form. And this oligomeric form is nothing but the clumsy or beta sheets of the alpha-synuclein, which is become a aggregates and eventually going to damage the varieties of macromolecules of the cells. In this case, I am interested, or my research group is interested on how this oligomer is going to affect on the mitochondria. Okay. So now I will going to show you this particular part, especially on the phosphorylation. Stay with me. Now, if you look at the very closely, this particular gene, which is having three regions, N-terminal, neck regions, and carboxylic end, this particular protein, which is on the post-translation modification, which is being regulated by varieties of PTM mechanism, which includes phosphorylation, truncations, acetylations, simulations, and nitrations. Among them, I am interested in only in the phosphorylations. And all these reactions are being carried by the varieties of, or it is being managed by varieties of enzymes. So I'm interested in a particular enzymes which drives the phosphorylation. And it is believed that there are varieties of amino acids on the different terminal, different part of the alpha-synuclein has been proved that their modifications play a great role in Parkinson's disease. But I'm mostly interested in S129 because S129 so far is believed to be the major culprit in case of the familial PD. So I'm going to show you a few of our new targets because the enzymes are now the targets. So I'll show you the real-time data, how we have found out this particular targets by using the multi-omics approach. Stay with me. Now, if you look at typically the alpha-synuclein, which is of now in the no more in the native form, which is of oligomeric in nature, which is in monomer, while comparing with the monomeric in nature, it is going to get transported into the mitochondria using the TOM complex. And the VDAX channels, once it is get internalized into the mitochondria, they perform their varieties of functions in order to maintain the mitophagy and fission and fusion process, which is again my downstream target, especially when I talk about the molecular mechanisms. What happened during the Parkinson disease, this oligomeric form is now no longer is allowed to be get inside into the mitochondria because TOM complex is going to get inhibited because it doesn't allow the the oligomeric form of the alpha-synuclein. However, the VDAC channel is still open and it is going to allow the oligomeric form of this alpha-synuclein which get internalized into the mitochondria, and it performs this varieties of dysfunctions which includes damaging the mitochondrial DNA. It is also going to get disturbs the fission and fusion process, and also going to get disturbs the mitophagy as well via the pink park independent ubiquitination. So these are all my targets, which I'm not going to focus on everyone, but I'm going to focus on one of the pathways which we have proved through the data. Now, so to find out those enzymes, what we have done, we have extracted the data series from the gene expression omnibus of 7 data series, out of the which the 94 from the control samples and 98 from the Parkinson's samples. And then on the next steps, we have extracted these data and we have done the image geoanalysis in order to find out the differentially expressed genes. What we have seen that among the normal and normal control and PD patients, there were nearly about 1,977 genes were found out to be differentially expressed. Among them, there were 980 was upregulated, 997 was downregulated. Since we were focused on kinases, was our target, especially the kinases are the one which is driving the phosphorylation reactions. So in order to find out the functional enrichment and annotations, we have given the molecular functions geoterminology, which is not limited to, but we have used 3 expanded terminology, which includes protein tyrosine kinase, transmembrane receptor protein kinase, and transmembrane receptor protein tyrosine kinase. So we have given the 3 geoterminology. Based on the 3 geoterminology, functional enrichment, which is called as based on the Panther classification system, we could be able to see that out of 1,977, there were 77 protein kinases which are involved in these number of genes. And among them, we have seen that there were 33 were upregulated and 39 were downregulated. So now we are interested in only in the upregulated kinases. Now, we wanted to see the interactions of these kinases with my target called as SNCA. So among them, when we interacted both the down and the upregulated kinases with the SNCA, we could be able to see that very specific kinases. See, kinase is a big family. Now to narrow down, we have found out the 6 new kinases. This is CSNK, MAPK1 and CLK4, CLK1, MKN2 and MAP4K4. So these are the 6 new kinases which has not been so far explored in the context of Parkinson's disease. Now, we wanted to check again the interaction of these 4 kinases with the SNCA using the bioeliminate software of serodangers, where we have done the H bonding and the salt bridging and pi stacking. Based on those, we could be able to see that they have shown a great amount of interactions. And then we wanted to see the interactions at the post-transcriptional level. This was the post-translational, and now we wanted to see the post-transcriptional because there are varieties of miRNAs which may play a great role in order to modify the alpha-synuclein at the post-transcription level as well. So in that case, we have performed the miRNet analysis using those 6 kinases with the SNCA, and we could be able to see that there were novel miRNAs which has not been so far discovered as a part of target for the Parkinson's disease. And they had shown a great interaction with the different kinases. So now we have 6 kinases to further validate these targets. So what we have done in the next step, we have took our SNCA protein structure, which we have drawn, and then we wanted to see the protein and the protein docking with the SNCA and all these most promising 4 kinases, which include CLK4, CLK1, CDK6, and MKNK2. But we have took additional very well-known ABL kinase, which is already there in the clinical trial at a phase 3 in cancer and many other chronic diseases. So we just wanted to check how my targets are behaving with respect to ABL, which is a very standard norm. And ABL kinase is also known to have a very great interaction with Parkinson's disease as well with respect to SNCA. And there are several compounds which has been trying in the clinical trials as well. So when we compare the Piper-Pose energy score and the Piper-Pose score, we could be able to see that the score was equivalent to what we have seen in case of ABL also. And that gives us the confident that yes, our target is not less than the standard one, and we wanted to go for the further analysis. So the next level of the work is going on. We have proved, or we are on the process of proving, that whether the CLK4, CLK1, and CDK6 and MKNK2 is really involved in the post-translation modifications of the alpha-synuclein or not. In that case, we are doing the CRISPR-based technology, and we are doing the knocking out of this particular kinases, and we wanted to further see the expressions of the SNCA is actually modifying or changing or not. So that work is right now going on. On the next step, this was the target base, unknown target. So we just wanted to find out the new target. In this case, the next study I'm talking about, in this case, I have a target fix, which is called as HP2X7 receptor, which is again a very well known in case of Parkinson's disease, and it is modulating varieties of neuroinflammatory pathways which are greatly involved in Parkinson's disease. So now I have a target fix, but I wanted to find out the new hit molecules. In this case, we have used multiple approaches. In this case, what we have done, we first created the homology modeling of our receptors, and then we have selected the ligand library selections using the different sources of Campbell and varieties of sort. Among them, we have selected only 3,521 phytoconstituents. However, the libraries are not limited to, there are varieties of libraries are there, you can choose those, but we have focused only on phytoconstituents. Based on the Lipinski rule, we could be able to shorten down or narrow down our 3,500 compound to the 2,142 compounds. And then 2,142 compounds has been gone through the virtual high throughput screening. Remember, this is called as virtual high throughput screening. And based on that, we could be able to narrow down to the 642 compounds. Now we have done the standard precision docking with womb of our receptors and our hit molecules of nothing but 642. We could be able to see that there were promising 192 compounds. And then we have done the extra precision docking. Among them, we could be able to see that we have seen only 57 compounds are showing the promising results. At the last, we have done the post-doc MMGBC assays, which is called as molecular mechanisms, the GBA score. We could be able to see that there are only 3 compounds was narrowed down from the 3,500. Now, the next challenge is to find out the next step of finding out whether these compounds are enough to perform their ADME profile and tox profile as well. So when we did the ADMET and the ToxNet using the ADMET software and the ToxNet software, we could be able to see that there are only 10 compounds were qualified from the 30 compounds. And then from those 10 compounds, we have went through the only 5 compounds based on the ADMET and tox profile. There are only 5 promising compounds, which we have right now with the work is going on, where we have done the molecular simulations, what is happening in the real time with those 5 compounds with my targets. So this work is not published, so I don't want to show my data because it's the open platform as a part of confidentiality. So this is how we perform the we utilize the bioinformatics approach in order to find out. I have shown 2 approaches. One is called as hit-based drug discovery, and one is called as target-based drug discovery. So there are plenty of other articles which we have published to understand the downstream mechanisms, which I have been talking about, especially on the autophagy pathway, which is of my, again, major target in terms of downstream, because this alpha-synuclein, which is formed during the Parkinson disease, it is going to go into the autophagosomes and is also going to damage the mitochondria, which you guys have seen in case of video. But it is not limited to the mitochondria. It does affects the varieties of mitochondrial motor proteins as well, which is helping the mitochondria for its retrograde and intrograde movements towards the the axonal tubules. So because since the neurons are highly energy demanding, so the mitochondrial movement is high in case of neuronal cells. It is get impaired in case of Parkinson's disease, especially the alpha-synuclein or oligomeric alpha-synuclein impairing the mitochondria and mitochondrial movement, also the motor protein, especially dynein, dynactin, and the KIF proteins. So one of my students wanted to see how one of the natural compounds called as bioclin could be able to restore the motor movement of these motor proteins using the mitochondria and improve the lysophagy and autophagy. I'm going to talk this in detail in the later slides. There are plenty of other articles which we recently published where we wanted to check the mitophagy pathway in great detail using the different natural compounds. Again, dimethylpurate has become our choice over the other molecules because of its already FDA approved molecule which is there in the market. We also have seen the autophagy in a great detail using the different pathway in case of Parkinson's disease also. And we have used another compound called as fingolimod because, as I discussed in the very initial slide, that the lipid dysregulation also play a great role. So in that case, we have again using the S1PR receptors where the fingolimod is already approved FDA drugs, which is used for the lipid lowering drugs. And this is going to play a great role in mitophagy also, which is a BNIP pink parking dependent pathway, which is again in my own downstream pathway, which I discussed initially in the pathology part. We also done certain other studies on fluoridine and autophagy pathway using the NRF mTOR pathway P62. And then I'm going to show you one of the, bioinformatics approach, which is already published article where we wanted to find out the network pharmacology, how the the toll-like receptor, which is of our again target in case of Parkinson's disease, because TLR is greatly involved in neuroinflammations. So it leads to the varieties of inflammatory pathways, eventually going to lead to the Parkinson's disease. So we wanted to find out the miRNA because, as I mentioned, that my two checkpoints are important as a part of my research. One is a post-transcriptional and post-translation. So this is a part of post-transcriptional because there are varieties of microRNAs are there, which is going to regulate varieties of the toll-like receptor signaling. So just wanted to show you, this is the graphical abstracts. Again, we have extracted the data series of Parkinson patients, among which there were 12 control samples and there were 12 normal samples. And then we have done the GOR analysis, and then we tried to do the differentially expressed genes, where we could be able to see there were 18,000 genes were found. We did the volcano plots and heat map analysis. And finally, we have did the gene enrichment based on the innate immunity, because our immunity was the main focus, where we could be able to see narrow down to a top 10 hub genes, and then we wanted to see those top 10 hub genes interactions with the varieties of microRNA, and then I'm going to show you in detail how we have performed. So what we have done, the differentially expressed genes which we have seen in case of volcano plot, if you could be able to see that they belongs to the varieties of pathways, molecular mechanisms. However, we were interested only in the immune systems, and when we narrow down the differentially expressed genes with the biological response, the different immunity, which is called as the immunity as a whole, then we focus on the innate immunity, the virus interactions, the apoptosis and adaptive immunities were majorly involved. The genes involved in those we have narrowed down, and we wanted to find out the top hub genes. On the next, if you could be able to see that when we perform the heat map analysis, comparing with the PD and control samples, the differentially expressed genes were high in case of PD, which is of pink in color. On the next step, we wanted to see the which pathway is highly belongs to the TLR signaling, where we have seen that toll-like receptors was found to be highly enriched in case of among all the pathways. And since we were interested in the immunity only, so we selected the top enrichment pathway, which is toll-like receptors. And then when you compare the toll-like receptors of only in immunity on all different types of toll-like receptors of downstream pathways, comparing with the control group and PD, we could be able to see that the TLR is highly expressed in case of heat map analysis of the Parkinson patient, which is shown in case of the pink color. So that gives us the confidence that just toll-like receptors of different pathways are involved in the Parkinson disease is not known, but we wanted to find out the novel interactions of toll-like receptor with the miRNA. And further, when we draw the interactions of the toll-like receptors, since we were focused on few pathways, using the KEC pathways, the TLR signaling representation associated with the NF-kappa B pathway, which is the majorly involved downstream pathway in the neuroinflammatory process, and the JAK stats were the major pathway that are downstream to almost all the TLRs, which we have been focusing on TLR 1, 2, 3, 4, and 5. Okay. And then finally, we made the protein-protein network analysis of the innate immunity gene expressed in Parkinson patients, comparing with the control. there was a huge interactions, which you guys can see. The total number of nodes were 314. And when we performed the top 10 hub genes interactions, it was showing a good amount of PPI network with the toll-like receptors, and that what we analyzed using the cytoscape software. And that gives us the promising and confidence, that the toll-like receptors involvement in the innate immunity is greatly. Further, we wanted to find out the miRNA network analysis, where we have done using the, again, miRNA database, where we have used the toll-like receptors and microRNA gene network analysis. Among them, we have selected top 5 miRNA regulating those TLR pathways of top 10 hub genes and the top 5 miRNAs. And now these miRNA we will be using as our target for modification at the post-transcription level as a part of our Parkinson disease modification of the toll-like receptors, which gives the neuroinflammation and induce the Parkinson disease. And when we see those top 5 miRNAs, by doing our differential pathological conditions and the gene network constructions using the cytoscapes, and when we see that the involvement of these miRNA is already reported in different disease, but not limited to, it also has a great role in case of the Alzheimer's disease as well. So that gives us again, the confidence that yes, the miRNA which we have been finding out is having a great role on different parts of the disease, not limited to, but also on the central immune system as well. So that gives us the lead idea about the precise targets that we can identify those miRNA, and further those miRNA can be used in case of, as a target point of in case of treating the Parkinson's disease, especially of the neuroinflammation associated with. Okay, so finally, I wanted to talk about one of the research what one of my students has done, where we know that the highly demanding or energy demanding neurons called as dopamine has a great kinetics movement of the mitochondria across the axons, because these mitochondria eventually involves in the release of the neurotransmitter at the terminal buttons. So the movement of these mitochondria across the axonal is called as retrograde transport. So this retrograde transport of the mitochondria, which you have seen in case of video also, being regulated by the 3 important protein called as motor adapter protein called as dynein, dynactin, and kinesin. So one of my students has believed that he made a hypothesis that the alpha-synuclein, which is going to damage the mitochondria, but at the same time also going to damage the motor proteins as well. So can we improve this using one of the hypotheses? And she designed this hypothesis that the bioiclin, which is known to have a great role in varieties of the other chronic diseases as well, and she had performed some in vitro. She planned to perform the in vitro assays that the bioiclin could be able to dissolve the beta sheets of the oligomeric form of the alpha-synuclein as well, and that's how it is going to improve not only the alpha-synuclein turnover, but also is going to improve the motor protein levels as well. And this he wanted to do by using the downstream pathway called as autophagosomes and lysophagy pathway. So I'll show you a few of the model system what she has adopted. So she took a C57B6 mice, and she has performed the intracerebroventricular injections of the rotenone, which mimics the Parkinson disease at the rate of 0.2 microliter per minute using the stereotactic surgery. And then she has performed the behavioral analysis of those animal post-injections, post-model development using the rotarod, which is the most gold standard method to understand the motor movement or grip strength of the animals, and also perform the actophotometer for the gross behavioral parameters. And then she has given the bioiclin for 2 doses level of 75 and 150 mg per kg for 28 days. And post-treatment, she has again performed the behavioral parameter, and then she compare the 0-day to 28-day parameters, and finally sacrifice the animal in order to understand the downstream pathways of those mechanisms she wanted to understand using varieties of biochemical parameters and immunohistochemistry. She also has validated her model by seeing the ipsilateral and contralateral movement of those animals. Oh, I don't know why this video is not 1 minute. Some problem. The video is supposed to be played. Anyway, so I wish, why not this video is not. So this is called as ipsilateral movement, where the animals are moving to the site of injections. This is called as ipsilateral movement, and that shows that yes, the animal has a great, damage into the brain. And then we have trained those animals. They remain on the revolving rod for almost 120 seconds of the cutoff time of about 300 seconds. And when we perform on the 36 days, these are the compromised animal of the rotenone treated. They shows the great amount of compromise in their motor movement. However, with the treatment of 28 days of the Biclean at the 75 and 150 mg per kg shows the great amount of the improvement in their motor movement. That shows that the animals are improving their Parkinson symptoms. We also performed the open field, where we have seen the distance traversed, mean velocity, rearing count, and resting count were also improved as comparing to the rotenone treatment group with the Biclean at the 75 and 150 mg per kg. And of course, when we talk about the target, these are the two important targets in case of Parkinson's disease. The tyrosine hydroxylase is the rate-limiting enzyme, which gives the idea about the turnover of the dopaminergic neurons, and alpha-synuclein, which is the culprit in case of Parkinson's disease. In case of disease control, the tyrosine hydroxylase was greatly decreased. On the other hand, the alpha-synuclein was overexpressed, which was got normalized with the treatment of different dose level of 75 mg and 150 mg per kg. This shows that yes, our drug is showing the promising results at the molecular level as well. And finally, to prove, we have done the immunohistochemistry, where we have stained our TH positive neurons with the primary and second antibody of the anti-TH. If you see, the rotenone treatment group has shown to decrease the dopaminergic neurons in the substantia nigra greatly, while in case of 75 mg and 150 mg of the Biclean could be able to restore these neurons, and that shows the promising outcome of our study that yes, our drug Biclean could be able to restore the end marker of the Parkinson's disease called as dopaminergic neurons. Further, we wanted to understand, since he was focused on the autophagy and lysophagy, he was focused on three important markers called as LAMP and LC3, which are the cytotoxic in nature. So this was got compromised in case of the Parkinson's disease, and then with the treatment of Biclean at the two different dose levels could be able to normalize it. However, the P62 got overexpressed because P62 is the marker for the autophagy, which is supposed to be decreased in case of the normal proteolytic process, but it has got improved in case of the Parkinson, which was got normalized with the treatment of Biclean. And then finally, she wants to prove her target protein called as dynein, dynactin, KIF, and dynein on the disease control got compromised as compared to the normal. When she has treated her animals with the 75 and 150 mg/kg of Biclean, it has got improved, and that shows that yes, the motor protein also got improved with respect to the improving the dopaminergic neurons and also decreasing the alpha-synuclein. And finally, she proved that yes, her drug, especially the Biclean, could be able to restore the alpha-synuclein and the motor movement of the across the. However, the limitations of the study was we wanted to perform the real-time in vitro assays of using the dopaminergic neurons and to check over the period of time of about 24 hours or 48 hours. We wanted to tag those mitochondria with our drug treatment. We wanted to see the movement of those motor proteins across the exons for on a time frame, which was the limitation of the study. However, the interesting outcome of the alpha beta tubulin ratio was not significant. We don't know what has happened to this, and we are still digging to find out what has happened to this ratio. There was no significant changes. And she finally concluded that yes, the impaired axonal transport was improved with the help of Biclean, and also they facilitate the autophagy as well, and they rescue the exon transport. That's how I'm going to finish with the time, as we are limited with the time. I have a few other studies to show you, the similar studies, which I don't want to go again because we have done the immunofluorescence study in other studies as well using dimethyl fumarates, using the dopaminergic neurons of the animals only, the NRF2 targets, and she wants to find out the autophagy regulations. She also done the varieties of the RT-PCR data as well of different proteins. Due to the limitation, I wanted to wind up my PPT. So just the conveying message is that the research is not limited to the basic science or the translation research, but there are other ideas that you can explore which is most exciting and innovative, I would like to share with you guys. My God, this video is not playing. Why this video is not coming? Okay. So this is the simple innovation that during the Parkinson disease, the patients are unable to perform their fine motor movements called as buttoning and then writing. So this is the very small innovation, but has a great impact on the lives of the suffering Parkinson patients. This is one of the innovative ideas which is not limited to the molecular science. Anybody can bring those ideas, and those are welcome to do, and they can translate those ideas to the suffering humanity. The other ideas is the spoon, which I have seen and I have experienced from my own hand. This was the anti-shaking spoon, just a small idea, and it could, serve the suffering society. I think now video is working, so I just wanted to show you the video of my animal study. Yes. Oops, still not working. Fine, so just wanted to end up with the thanks to everyone and would like to take few questions and of course would like to acknowledge my whole team who have been generating those data. Most of them are in industry, some of them are pursuing PhD, and she is the one who has developed the data, especially of PD, the Biclin one. She's now in Germany, she's doing her own PhDs, and some of them are PhD students. I would like to thank all of my students who have been doing the hard work in order to develop those data. Yes.
It was nothing sort of exceptional, sir. We totally acknowledge the time and you have given time, energy you have given to this research, and definitely it was a brainstorming session for us. We'll definitely go through your previous research works so that we could also get more in-depth detail about this. Sir, in last we have received questions, but out of them I have filtered some of. So one of the question is, what recent advancement in genomics, proteomics, metabolomics, and lipidomics have significantly impacted drug discovery?
If we look at conventionally and what we have been hearing from our textbooks, that the 90% of the drugs, the clinical trial fails at the later stage of clinical trials. When it comes to especially in the phase 2. The reason because we don't have a refined targets, and we don't have precise targets, and we don't have refined lead molecules. So utilizing those approaches, especially of this called as network pharmacology, when we utilize those proteomics, genomics, and it's called as whole it's called as whole genomics, whole proteomics. So if we implement those technologies, we could actually prevent the loss of those the later stage of clinical trials, the later stage of drug discovery and the clinical trials. So we don't have to wait for that much of time, where utilizing those platforms, we can actually refine our molecules, we can actually refine our targets for a precision based drug discovery. So yes, of course, since it is emerging technologies which people are now being adopting it, so this will give us the idea about the most promising targets and most promising hit molecules.
Right, sir. Thank you so much for this. And we have next question, that how are regulatory agencies responding to the integration of multiomics in drug development, and what guidelines are emerging?
So far, I believe that there is no guideline as such as a part of the research to adopt these technologies. You can perform your data as far as When the regulatory comes, I don't think so far there has been any implementations or regulations on utilizing the proteomics approach. If you look at, especially from the Indian context, when it comes to the submission of your CD documents, called as common technical documents, where you have varieties of, 1, 2, 3, and 4. The 4 is specifically for the data which we are producing in case of preclinical studies. So everything depends on the 3 important parameters, that on every stage of your drug discovery steps, you have to see the 3 important parameter. One is called as, the very first is safety, second is toxicity, and third is efficacy. If you are proving your drug molecules on every step of your drug discovery that your drug is promising, you can utilize any approach. However, those approach has nothing to do with the regulatory, as a part of how you are generating data, whether it is approved from the regulatory agency or as per the requirement of regulatory. Which instrument which you are using or which platform which you are using is not has came so far in the regulatory context as a part of regulating those, to the best of my knowledge.
Thank you, sir. We have a last question, then we will conclude for sure. And the question is, what real-world applications of multiomics in drug discovery has shown the most promise in recent years?
Most promising outcome, I would say that they wanted to find out the most promising results.
Yes,
In the context of my own field, I would say that in the case of Parkinson disease, because I follow many of the scientists across the world, and especially I would like to mention here the name called as the Dawson husband and wife, who are the pioneer in doing this research, and the other one is the scientist name is called from Iran. So these are the 2 pioneers who have been utilizing these approaches, especially to understand the alpha-synuclein from the multi-omics approach, which called as proteomics, because I am focused on proteomics. So they're utilizing these approaches in order to refine whether the involvement of those alpha-synuclein is really or not, and if it is, then which pathway they are following, which protein they are modifying. Yes, there are been promising results which in the context of Parkinson disease, and there have been a great advancement has been done with respect to Parkinson disease. When I talk about the super specializations, you can find out on the Michael J. Fox, which is one of the organizations who is a pioneering and who is supporting the Parkinson disease research, where there have been plenty of research has been done utilizing those platforms to find out the refined targets.
Well, thank you so much, sir. As we conclude our 55th SPSR webinar on multi-omics approach, a paradigm shift in drug discovery, it is clear that we stand as the cusp of transformative era in biomedical research. The integration of multi-omics technologies encompassing genomics, proteomics, metabolomics, and lipidomics has fundamentally redefined our understanding of biological system and disease mechanisms. Throughout this webinar, our distinguished speaker have illuminated how multi-omics approaches are revolutionizing the drug discovery landscape by providing a more detailed, interconnected understanding of molecular underpinning of disease. These technologies are uncovering biomarkers, identifying new therapeutic targets. So at the end, we are truly deeply thankful to Dr. Shashi Alok sir, the National President of SPSR, for honoring us with his presence. We recognize and appreciate your commitment and guidance for you. We are truly thankful to contribution of our keynote speaker, Dr. Dharmendra Kumar Khatri sir, whose expertise provided us with the valuable understanding of multiomics approach, a paradigm shift in drug discovery. We also acknowledge our convener, Mrs. Monika Sabharwal ma'am, for a critical role in organizing this event. Our gratitude extends to all the participants from all corners of the world for their joint effort, which has expanded our knowledge and encouraged cooperative learning. We are grateful for the significant involvement in 55th SPSR webinar. Your engagement, participation, and input has essential and anticipate collaboration on upcoming initiatives enhance healthcare and well-being. Upcoming 56th SPSR webinar on innovative multifunctional dressings of accelerated infected wound healing, nuances and achievements from IPK labs will be, the esteemed speaker will be Dr. Indupal Kaur ma'am. So please do share, subscribe to the official YouTube channel of SPSR for such brainstorming webinars. Thank you so much. Have a wonderful day ahead. Thank you so much, sir. Once again, it's our pleasure.
Thank you, everyone.
It was our pleasure, sir.
Hope you enjoy.